please join me in welcoming Peter Lidsky as our second speaker, who's going to talk to us about how aging could be a kin-selected pathogen control mechanism. Welcome.
Thank you very much for having me here. Big proud and pleasure. So the title of, so I'm Peter Lidsky. I'm an incoming professor at City University of Hong Kong. And so the title is Why Do We Age and How to Stop It? Because as you probably all realized, we'll all age and we need to do something about it. And the first idea that is around in literature, we need to eat less. So if we starve worms, then they live much longer. And this brings us to sort of conclusion that the amount of food the organism can consume per lifespan is sort of constant. The question is how do you, use this limit? And if you go to some general conference, like this Drosophila Research Conference, and the pointer started working at this point, so we find that aging is grouped with physiology, metabolism, nutrition, this cloud of ideas. However, the funny thing is, if we give worms to smell food, but don't give them opportunity to eat it, then effect is dramatically reduced. So they live shorter than if we wouldn't give them this smell. And there are neurons in the brains of the worm. If we ablate them, then the effect disappears and they again live longer. So there is something what modulates aging. Excellent. Something that modulates aging that comes through the brain, right? So we don't need to eat much. We need to sniff things carefully. But what about looking at things? Can we stare safely? Apparently not, specifically if we are Drosophila. So if we are Drosophila and we stare on the corpses of other Drosophila, then we accelerate aging. And there are also neurons identified in the brain of Drosophila. If we ablate them, kill them, then the effect disappears. So, question is where we are, what does it all mean, what is aging? And if we ask different people, apparently, surprisingly, we realize that we don't know what aging is. So this is a big collection of top-notch experts, and they were asked what aging is. They just came to the lack of consensus and agreement to disagree. So aging is one of the last entities, big questions that doesn't have a paradigm. And we need to look at aging like if we would look at infections before Louis Pasteur's discovery of germ theory. And this is a very unusual look for today because most of us grew up in very mature, established fields with the paradigms designed 70 years ago. And working in the field without paradigm is a skill that is almost missing in other parts of biology. So why do we need a paradigm? What the new paradigm should define? The first question is aging like entropic process, damage accumulation, or it is a programmed process? So this dichotomy defines what we do. So if aging is an entropic process, then we need to classify all these damages, make these dots to fly out slowly or turn them back into their places. This is Aubrey de Grey's approach, classify all the damages, make a little pill against every damage, and give them to patients. And in case aging is programmed, then we need to find this gun and remove the bullets. So in this case, we need to repair something. And in this case, we need to break something down. And these are two completely different approaches. They can have a little overlap, but most probably if we choose in the wrong direction, 95% of resources will be lost. So, and currently, 100% of people pursue this entropic vision. And so my approach is to look in nature and see what we see there, and we see surprising things. For example, very similar animals like rats and squirrels have very different lifespans. So rats can Harold Ketcher's showed that rats can survive a bit longer, but the average lifespan of a rat is 2 years, but for squirrels, they can live up to 20. So, and why does it happen? Traditional take of it is that the efficiency of damage control is adjusted to particular ecological niche, and it is explained by some boring factors like extrinsic mortality, maturation time. So, and if you talk to people in the field, they all will say that this is like boring, established, and just very well characterized. The touchy moment is that it is not. So if you look at some long-lived animals, some of them have very fast maturation speed, and some of them have very fast extrinsic mortality. So the correlation between these ecologic factors that are often cited as ecologic determinants of aging is not perfect. And moreover, if we speak about age-independent extrinsic mortality, so death of hunger, predation, etc. So it should not affect evolution of aging according to the, this is from demographers, this is from ecologists. So even theoretically, there should not be a link. So coming back to our rat and squirrel, this is only part of the gradient. So on the long-lived side of a gradient, this is naked mole rat, an animal that barely age at all. And Rachel Buffenstein will be telling in more detail about it. And on the other side of a gradient, there are these semiparous animals. This is a northern quoll, a carnivorous marsupial, that actually, what is semiparity? It's a die-off after reproduction. So it happens in males. So after mating, after breeding season, they all like sharply die because of hormonal effects. Like they lose fur and cortisol levels are peaking, and like 100% of males die. So interestingly, the similarity evolved in marsupials 5 times independently, and only in carnivorous marsupials, not in any other mammals. So a classic example of similarity is, of course, the Pacific salmon, a fish that goes up the river and dies. But in mammals, it's found only in carnivorous marsupials. Why? Why are mole rats so non-aging? What ecological factors explain that? No answer. So, and where is damage in this equation? It's difficult to rationalize. Of course, if we take old patients, old animals, we see damages everywhere, but the same way we can find damages in all other diseases. If you have a diabetic person, late stage, we will find a lot of damage in his body. If we take infection, if it's cystic fibrosis, we always find damage, but only in aging we think that attacking this damage would be of some help. So, and coming back to the animals, how do we understand what's going on? So the only way to understand things in biology is evolution. Nothing in biology makes sense except in the light of evolution. Theodoros Dobzhansky, classical quote. So our bodies are made to, so evolved to spread our genes, and this gives biology this idea of a purpose that helps us a lot to rationalize things. And what is the evolutionary take of aging? Josh already explained some of it, so I will probably repeat in a little bit more detail. So the first idea was, this came from Medawar and Haldane. It's called selection shadow theory, that animals die of predation, hunger, etc. And if nobody is surviving to this stage, then what happens here doesn't really matter, right? So because nobody survives, so fitness cost of aging here wouldn't be important at all. So, but this model predicts that animals should not age in the wild, and this is not true. This is not true. So if we look at a wild population of animals, let's say lions of age of 14, you see in wild populations, this is mortality. mortality is much higher than in lions of age of 4. And lions survive to the age of 14 in the wild. So you see it grows. So this theory is incomplete. Of course, if we have like a sharp deterministic mortality, let's say explained by seasonal changes, like in this killifish that Dario will probably tell us more later on. So fishes from ephemeral ponds that dry out during dry season in Africa, they all die. And then it's indeed nobody is surviving to the age, and then it's indeed not important what happens there. But this is a very unique situation. In most animals, individuals survive to the age of aging. So, and this was first realized by this guy, George Williams, who introduced his antagonistic pleiotropy to complement selection shadow idea. Idea is that there are pleiotropic genes which are good in our young years and bad in our old years. And since still this extrinsic mortality is there, what happens in our young years is much more important for evolution than what happens in old years, because this cohort is smaller. And if we accept the existence of pleiotropic genes, then it can work. The problem is that this implies non-involvable features. So there are two different properties of the genes that evolution should not be able to disconnect. And this is impossible to imagine even theoretically. So I haven't found a single, even realistic example of pleiotropic genes. So another conceptual upgrade for the theory has been proposed by Kirkwood, that was already mentioned by Josh. So his idea was that aging is energy-saving mechanism, that we have limited budget, and the budget we can invest either in reproduction, either in repair. If we invest in repair, then we live longer, but reproduce less. Right? And this, on the left side, we invested in reproduction. The parents died, but by the time of 2T, here we have 4 animals, here we have 3 animals. And these agers with this trick can overpopulate, overgrow, overreplicate the slow ager. So the problem was already mentioned by Josh. So the caloric restriction doesn't fit at all, and there are much more things that contradict this theory. That, for example, males are aging faster than females while investing much less in reproduction. Food consumption decreases with the age, right? Well, logically, it should be like if an old animal found enough food, it should eat more and either rejuvenate or prolong its lifespan. Never happens. Poor ecosystems that should stimulate the food energy saving, like Arctic caves, deep water, they all are full of centenarian species. And it clearly shows that longevity is much more energy efficient strategy than replication. So, and coming back again to the choice between damaged and programmed models. So in the end, if we follow the evolution of the ideas, our focus on damage accumulation theories comes to these very ambiguous models. But why don't we believe that, no, sorry, we do, but why most people in the field don't believe programmed aging can be there? There are two very good arguments. It's not clear what this program can be good for. And second, why don't we see mortal mutants? So, and let's now examine these questions. First of all, are there examples of programmed death in nature? Yeah, there are such examples. For example, bacteria infected with a phage tries to kill itself to prevent phage replication. So if it does it early, then the phage dies with this bacteria, and the neighboring bacteria who are related to this one, they are not getting infected. So, and this is clearly part of the immune system. Phage is trying to inhibit it. So the bacteria is trying to overcome this inhibition, kill itself in other ways. So this is like a typical immune coevolution, tug of war between host and pathogen. So, and if it is, so there is clearly example, but can it be true also for aging? So can it be that like pathogens, and if aging is something like that, immune adaptation, then what would be the predictions? First of all, pathogens should prolong lifespan, which is sort of paradoxical. And second, hosts should reduce lifespan if infected. So this would be the, if aging is needed to limit pathogens somehow, then hosts will be willing to age faster, and pathogens will be willing to make us living longer. Other examples of pathogens that prolong lifespan? Apparently, yes. So, and this study showed that in ants, ants, they have queens and workers. And queens are living like 50 times longer than workers, while it is the same genome. It's just two different programs. And some tapeworms are taking advantage of it and switch workers' longevity to a queen style. And this yellow line, I'm sorry about the color scheme. I just copy-pasted from the paper. The yellow line is here. Green is queen's survival. This is workers' survival. So you see that infected workers are living the same, aging the same speed as queens. So, and this, for chronic pathogen, host longevity is advantageous. This is found in every mathematical model, and it is logically correct. Why these examples are so scarce? I think because for this we should have some switch between two programs. So pathogens probably cannot do like humans to grow wings, right? But if there is some genetic predisposition, some alternative program that can be switched, then pathogens can exploit that. So at least one of the prediction, sort of in some cases at least, can work. Second, does acceleration of aging, does host accelerate their aging by infection? It seems to be so. Let's say in HIV-infected patients, we see that aging is accelerated by Steve Horvath's these methylation clocks, and we can reproduce it in Drosophila. So if we express replicon, this is a piece of a virus that can replicate and induce immune system, but cannot produce virus particles. We see that these flies are dying faster, and by transcriptomic clocks, they also age faster. So this is like aging baseline, like flies collected 14, 21, etc. days. And this is just PC1 component that is responsible for the age. And you see that replicon expressing flies that have this virus simulation, they are older than controls that have inactive replicon. So it seemed to be that indeed hosts accelerate aging in response to infection. And this is consistent with all the data about inflammation. Every inflammation is very bad for the aging speed. So it seems to be both observations can be somehow validated. Now, what is the mechanism of selection? So let's come to this fish from Norwegian lake, and this is its parasite. If we collect fish of different ages, we find that the prevalence of parasites is increasing, and by the age of 8, almost every fish is infected. This parasite doesn't sterilize fish, but let's say it would do it. So if this fish cannot reproduce because of the infection, then they are dead for evolution. They will anyway die at some point without leaving any progeny. But they can infect their relatives that swim nearby. And this makes this fish actually toxic for their own genes. And then after this age, evolution may want to remove every animal just based on age, just by the statistical expectation, not by any other reason. So, but the particular moment that is around in all that logic that these neighbors should live nearby. So the relative organisms should infect each other with higher probability than non-related organisms. And this is just technically, it's difficult to model, and this effect hasn't been studied that much. So my main contribution to the field of mathematical modeling is this model of infections in viscous population. In this viscous population, every agent is an animal, so they move on the plate and reject each other, like charged balls on the surface of dielectric. So you see their tracks. They remind this GPS tracked wolf in Minnesota. And this model recapitulates two very general properties of animal populations that are typically overlooked by regular epidemiological modeling. So in particular, there is this kinship gradient that relatives interact more often than non-relatives. And second, there is density-dependent dispersal. So the animals from densely populated areas migrate to the more sparsely populated areas in the search for better life. Both effects are quite general. And now we run in epidemics of sterilizing disease in this model. So if these are short-lived animals, these are long-lived animals. So, and you see that in short-lived animals, epidemics goes at much lower level because this is chronic disease that animals cannot recuperate. So, and because the old animals are removed, so the epidemics goes at lower level. And if we, without infection, introduce a long-lived mutant into this short-lived population, of course it takes over because it lives longer, replicates better, and it's more successful. However, if we do it in the presence of this chronic sterilizing pathogen, situation changes. So you see that the infection goes much more efficient in this long-lived variant. Animals get infected at higher rates, sterilized, and expelled from the population. And this model can explain why, so like a set point of the lifespan ecologically, and as you might recognize, I am George Mitteldorf 20 years earlier, because my theory is only 7 years old. But this model doesn't require group selection. So it can be run only on kin selection, whatever. I'm sorry, I don't have time to explain for people who don't know this big debate between evolutionary biology. So with this model, we explained what aging is good for. But what the heck, why don't we see mortal mutants? If we take our flies or worms and we treat them with mutagens, we immediately find flies without wings, without eyes, whatever mutants, but we never see ageless flies, flies that don't age. And if aging is programmed, then there should be genes that execute aging, right? And then we need to delete these genes and get flies without aging, but we never see it. And this is considered a killer argument against every aging theory. Repeating, so we see what can be called gain of function mutant, but we never see aging loss of function mutant. But this is not true. So the loss of function mutant must exist because naked mole rat that basically doesn't age that much has common ancestor with other rodents. So at some point along this branch, an aging mother should have produced a non-aging child. So immortal mutant must exist. They are just very, very rare. And the question is why? And my answer that my theory produces is that because of these pathogens are trying to hijack aging and extend host lifespan. So, and to defend aging against this manipulation, evolution developed some security belts that makes aging so evolutionarily stable that it is resistant to pathogens manipulation and also to mutations and to, unfortunately for us, to therapies. So, and with this theory, I cannot go much in detail. In details, we this is from my review. So we classified all the all the observations, and these are all major theories, and our pathogen control theory can explain much more than any other. So including this, these phenotypes basically are not explained by classic models. So why everything what flies lives so long, why mole rats are non-aging, why this difference between eusocial insect case longevity and also the similparity. But it explains things better, but still, this is like a very big claim. So because it should reclassify aging from this physiology metabolism group of things into this immunity microbiome infection group of things. And of course, when you come with these two people, they, so I understand them. If I would be like studying my viruses and somebody come and say, hey, Peter, I discovered aging. Do you want to read it? What I would answer. So, yeah, very interesting, but you know, I am on the run now because it's 100% psychopath, right? So, and this is my life. Yeah, so obviously everybody in science work looking in their plates, and to make people to raise their eyes, you need to produce some groundbreaking, something mind-blowing. So, and I have two ideas what mind-blowing can be produced. The model has two predictions. One concerns naked mole rats. So why can mole rats live so long, like ecologic reasons? So there is an alternative way how to control infection, the same as found in bacteria. So if the very first animal that contacts disease dies, kills itself, this is a harakiri knife. If it dies by adaptive suicide immediately after infection, then we can keep population actually sterile, not in terms of reproduction, just there will be no pathogens at all, and this long-lived invader can indeed overcome the short-lived animals. But unfortunately, this strategy is available only, probably is associated with eusociality. Because in eusociality, you know, mole rats are eusocial, meaning that they have queens like insects, ants, and workers that don't reproduce. And in this situation, a worker's suicide will be disconnected from the reproduction, and worker's killing self will save the queen and increase the worker's reproductive fitness. So, and this can be like a testable prediction that we can do I don't have mole rats, unfortunately. I need collaborations. But so there are like actually two papers that indicate that mole rats indeed can have this phenotype, so they are super susceptible to herpes simplex virus and seem to be very susceptible to coronavirus. And this just need to be tested at a better level. And the second groundbreaking, the mind-blowing result I think can be done concerns this similparity and repeated evolution of similparity in marsupials. So my model predicts there should be some unique disease running in this population. And if you look at the literature, we find this disease. So this is this facial tumor of Tasmanian devils. Tasmanian devils are not semelparous. They are iteroparous right now, that they reproduce several times without dying, but they are partially semelparous. They males lose weight, and there is like anemia, and they are like somewhere in between. And this is the biggest representative of these carnivorous marsupial groups. So, and the disease is actually transmissible cancer, and the infectious agent, the pathogen, are cells of the Tasmanian devil that died 30 years ago. So these are cells from a female that developed this cancer first. They are spread across a population of devils, killing most of the animals. So this is a very unique disease for mammals, but surprisingly, in Tasmanian devils, there are two independent lines identified. One came from a female 30 years ago, another from a male like 20 years ago. So like half of all described lines of transmissible cancers are found in this species, which is very surprising. It seems to be they are prone to this induction of these cancers. And then the genius student that worked with me, we built a model that, for this disease, and we find that this disease indeed can drive evolution of similparity. So idea is that it is a very transmissible disease, very lethal, but with a long incubation period. So it peaks during the breeding season, and the male that survived the breeding season is 100% likely infected. It will not make it to the next breeding season, but will roam around and infect its potential relatives. And then it's better for this male to die straight away after reproduction. So this is our model outcome. So blue animals are like this similparous strain with male similparity. With no disease, of course, it dies out very rapidly. But if we have this DFTD disease roaming around, then similparous variant can win to some extent. So it doesn't mean that devils are on the way to evolve similparity, but under other population structures, it can happen. And the physiological model, why are marsupials so susceptible to induce these transmissible cancers, so that they don't have placenta, but the embryo is attached to the uterus in order to feed. And PD-L1 protein in our bodies is responsible for protection of the placenta and embryo against the mother's immune response. And marsupials don't have that. So the attachment of the embryo induce inflammation. And therefore, these animals, marsupials, can be in a trade-off between length of embryo attachment and feeding and non-self-recognition. And indeed, there are small papers that indeed some marsupials indeed miss these parts of this non-self-recognition. So, and my idea is that that makes marsupials more susceptible to transmissible cancers induction, and this can explain persistence of transmissible cancers that in turn drive evolution of similparity. Idea would be just to catch some marsupials and look at their tumors if they derived from different animals, just by genomic tools. So these are two predictions to test in collaborations: hypersensitivity in mole rats, transmissible cancers in marsupials. So conclusion number one, aging may be an adaptive process, evolved to remove old and potentially infected individuals. And what are the mechanistic predictions? Okay, if aging is a functional part of the immune system, it should be tightly interconnected with other immune mechanisms. Then, actually, rejuvenation of the immune system should be a good direction to go. And Greg, I think, will tell more about it later. So I want to report our work on cellular senescence. So what is cellular senescence? So there is this organization called ICSA. So this is from their website. So cellular senescence is a programmed state of stable cell arrest that is accompanied by a complex phenotype. Senescent cells play a role in physiological processes such as tumor suppression, wound healing, and embryonic development, whilst paradoxically, they can contribute to aging, cancer, and age-related diseases. As such, the field of cellular senescence represents a multidisciplinary research topic. Not very clear what's going on here. So the idea is that in normal tissues, so I just partially repeat what Josh already told. In response to DNA damage, telomere shortening, or virus infection, some cells can become senescent. Then they do this secretion, and with this secretion, then they can make other cells to like a secondary senescent state. And accumulation of the senescent cells leads to metabolic disorder and cancers and other age-related diseases. So initially, senescent cells were considered as anti-cancer thing because it develops in response to telomere shortening, cell divides, telomeres are getting shorter, then it cannot divide anymore, goes to senescence, cleaned by immune system, and also this secretion has been considered as like something that can attract immune cells and help to resolve this cancerous situation. However, later appeared that this persistence of immune cells and their secretion is actually good for cancer, very bad for patient outcome. So every aspect of cancer is increased by senescent cells. In stemness, proliferation, angiogenesis, and the vessels are growing into the tumor, which is needed for its nutrition. Also metastasis and immune evasion, all that is stimulated by secretion of senescent cells. And it is not clear why did they evolve. And if you look of what is activated in senescent cells, as a virologist, I see all my best friends innate immune molecules that actually are activated in senescent cells. And like 2016, there was a paper published by a Spanish group, published in a relatively small journal that showed that senescent cells are antiviral basically. That if you infect these senescent cell lines and infect with GFP-labeled viruses replicate to lower level. The results were there, but they were not that strong. So we taken advantage. So this is a student who used to work with me. So we did this. So these cells are controls, A549s. These are senescent A549s. Would be everything green. Here is very little. So the senescent cells are antiviral. And funny thing is that senescent cells are very diverse. It's not like black and white, senescent, not senescent. There is like complex phenotypes, and they are different, and this is like process developed with time. And with different inductions, even induction with the same drug at different concentration, different days, we can see the different phenotypes in antiviral activity. So see, this is beta-gal staining, which is marker of cellular senescence. You see that all these cells are senescent, but if you look at antiviral activity, these are viral titers. These are controls, polio, mango, vesicular stomatitis, Synbis virus, Zika virus. They all are inhibited in senescent cells. But in this probe, by some reason, the antiviral activity is much lower. So, and we used it just as a tool to figure out what's going on. We did RNA-seq bulk. So just on control cells induced by these different inductions. And we see that there are like 25 transcripts, 25 thousands of transcripts changing. Best luck to find what it is. However, and you see the clusterization is okay. These two probes that were induced by bleomycin cluster together, doxorubicin induction cluster a little bit away, so control, so the clustering is consistent with what has been done. But if we look at this now, how to figure out what is the antiviral activity, we look at the cluster in which antiviral activity is high. So we basically look at the genes that correlate with antiviral activity. So these genes are not activated in this BLEO8 probe, which has low antiviral activity. And what we find only less than 300 transcripts. We do string analysis. We find all, so they form a cluster, immune system interference-stimulated genes. So, and indeed, the media from senescent cells can inhibit replication in healthy cells. So if we put these secretes from senescent cells, put on healthy cells, then viruses don't like it. So, and indeed, secretomics, senescent cells produce much more cytokines, just in control and if infected. So this is still ongoing, and this JAK-STAT pathway, which is downstream from interferon signaling, is, if we inhibit it with a drug, this has replication of vesicular stomatitis virus in senescent cells, and this is with a drug, so it is recovered to the wild type level. So at least in under these conditions, it is because of interference. So they do interference, excite them. So next is that this, if we knock out either interferon receptor or this JAK-STAT pathway component, we see that the replication increases if compared to control. So it's clearly something about interferon, at least. It doesn't mean that it's 100%, but this is involved. So my working model is that senescent cells play a role of this immune militia. So there are immune cells, which are professional soldiers, and there are militiamen that can be recruited from basically every other job. And these militiamen, so they can be induced by viruses. They produce this SASP. They can activate other cells to become more protective or also become militiamen. And also they communicate with professional soldiers. And I believe this is a primary function of senescent cells that was later repurposed for aging. So with this, I want to thank people I was happy to work with. So my ex-boss, Raul Andino, these two students who worked with me. This year, one got to Yale and another to Harvard. So as a commenter, I collected the Flash Royale this year. And I myself go to the City University of Hong Kong, and I'm hiring. If you are interested, reach out to me. Questions?
Hey Peter, I really loved your talk. Thanks so much. This was super fun. I have several questions. So, maybe I can start from the last part. And maybe I missed that. I really understand the concept of senescent cells as sort of like this antiviral militia. But what happens at organismal level, whether the immune cells themselves are senescent?
Yeah. This is a very good question. So we have an evolutionary model behind that. So, but I believe that initial function of senescent cells is this militia, but then they got repurposed to kill ourselves also as a side project. And in this, by making immune cells senescent, we change the functionality of the immune system in order to kill ourselves. I don't really have, I don't think we have a time to go into details about the evolutionary reasons, but we can talk about it later.
Yeah, no, I mean in the context of, broadly speaking, kind of like this immune militia. But yeah, I would understand that for non-immune cells, if the senescent cells are the non-immune cells, then I can totally see, because you sort of dampen those immune responses that might be detrimental at the level.
So the immune system is one of the experience the strongest changes during the life. And it according to my evolutionary model, it should become less protective against new diseases and more protective against diseases that organisms encounter earlier. So, and with, I really don't have time to explain all the epidemiological things. For who is interested can come. But I think the senescence in the immune system assists this role, so that the new pathogens should kill older individuals. Old pathogens should be okay.
So okay, I'd be curious to definitely know more about it. Can I ask a second question?
Yes, please do.
So regarding your first part about this sort of kin selection and the fact that aging at the population level can sort of prevent disease spread, right? So this is pretty much, which is very similar to what also Josh worked at, like in 2009. There is a brilliant paper, Josh, Medawar, and Pepper, I think, from
this site.
So where they also proposed something very similar, but it was not kin selection, if I'm not mistaken. That was more global, population-wide effect. So my question to you is, I see that your model works not necessarily at the level of aging, but at the level of longevity, right? Because as David mentioned at the beginning, aging has to do with this kind of age-dependent increase. Oftentimes at the population level is described as exponential increase in the probability of that, right? In your case, you don't need aging really. What you're really talking here about is longevity, survivorship, which could be age completely independent. Could be the baseline mortality that you can even have a non-aging population, meaning probability of death at each age is constant, which leads to an exponential mortality still in the population. But if you instead of having a 0.9% survivorship at age, you have 0.8, but then you still have an exponential.
Of course, the age-independent mortality is playing a role there, so it can.
But it's not necessary for your model, right? So because what you're tweaking with your model is actually turnover, basically, right? So you don't need aging.
No, there's not turnover.
Population turnover.
First of all, I think if you use word population in this context, some evolutionary biology would like to burn you out of that because it's group selection. So it is kin selection. So we are thinking about how individual helps its relatives to avoid infection. And so even if there is very small amount of animals that survive to a given age, these animals can be toxic for their
But you don't need aging.
Huh?
You don't need aging.
No, but you don't need aging if nobody survives to the age when it is almost 100%.
Yeah, but exactly, right? So what you're talking about is not aging, it's longevity, right?
Yeah,
so it should be aging.
But just aging is something that ensures the longevity. So
No, not really, because you can have, that's what I mean. You can have constant age mortality, which leads to exponential decline in survivorship at the individual level.
Yeah, but this is probabilistic process, right?
Yeah,
so, but it means that the number of animals surviving to the age X will never be 0, right? So this is like a rule of math, and let's say if animals at the age of X are toxic, then evolution need to take care, and survival is not 0, then evolution should take care of these animals and remove them by aging. So, of course, there is this. I see your argument, but this is a quantitative argument. So, you can reach you can reach neutrality only by 100% killing.
I'm not saying it's a wrong argument. I'm simply saying that you don't need aging for your argument. That you just control longevity at the individual level, and then you have the pathogen clearance. But it's different from aging.
No. How do I control at individual level age-dependent mortality? So
you have baseline mortality. Anyway, we can talk offline.
Baseline mortality is probabilistic, so you will never have by the age of X, you always, with probabilistic process, you will always have non-zero number of individuals.
A number dependent, yeah.
Yeah, it's number dependent, but if you integrate over time, you always have non-zero number.
But you have a very low density, so you decrease by, in your kin, not in the population, you will spread of the disease. So it effectively works at the same time.
So there are, of course, I agree that this parameter is important for the model to work, but it doesn't remove the necessity to remove older individuals. So sooner or later you should do it, and actually this can explain this. I did modeling yet. So this debate about is extrinsic mortality prolonging lifespan or shortening lifespan evolutionary. This of course, Resnick's paper, right? Yeah, so that it can be that in some cases extrinsic mortality can indeed replace aging. David Resnick, yeah, guppies, right? Yeah. So Josh wants to say something. I just want to explain for people who don't know. The Resnick collected guppies from different streams, some with predators, some without predators, and he found that guppies from the streams with predators were living longer than predator-free guppies, which was opposite to the mainstream thinking at that time.
Not really. You didn't find differences in survivorship, right? The only difference that you found, if you refer to the science paper,
I think aging was slower.
There was only one thing that was behavior. that was different? That was
I
start
think
to
it was
survivorship wasn't.
Oh yeah, survivorship wasn't effective. Yeah, you're right.
He was he's coming with a very strong expectation because all the predictions from Williams were sort of like not met by this perfect, seemingly perfect.
But I think they were aging slower, so they are swimming better at older age.
Exactly, they had one difference in the behavior, but besides that,
I think it was physiology, not
yeah, but behavior is integrated.
but definitely, so extrinsic mortality can modulate the stuff in both ways, probably, and here I agree. But you never come to with stochastic killing. You will never come to a situation you don't need it. That's my stance. Yeah, George.
Sir, thank you. I've been in this field for so long, it's hard for me to get new ideas, and you've filled me with new ideas to consider, which is, I'm very grateful and congratulate you. The thing that I'm able to respond to most quickly is the evolutionary simulations based on infection, because I've done some work in that area too. And so quite a couple of questions come to mind. One is, in your model, the newborn animals are all uninfected?
Yes.
So did you try, what happens if the infection of the old animal is...
So if
did you try that as a variant where infection is passed on to the offspring?
Oh yeah. So the
does that wreck the model?
Huh?
Does that ruin the model?
No, it makes it stronger.
It still works?
It makes it stronger. It makes it stronger because it is like addition to the sterilization effect of the pathogen. So like...
this is a sterilizing pathogen.
The sterilizing, oh, I didn't tell that. So it was written in red lines.
It's a non-sterilizing pathogen?
No, non-sterilizing pathogen do not work anything.
It doesn't work at all?
Yeah, no, so obviously I didn't explain it very well, but just the idea is that pathogens, chronic pathogens make host weak, and this host cannot reproduce very well. So yeah, these are sterilizing pathogens. Any more questions?
Question. Yeah, it's very interesting talk. Thank you. It seems like the evolutionary optimum thing to do would be to sense, for the organism to sense that a pathogen is taking hold and kind of self-destruct very rapidly, right? How do you explain the gradual, progression of aging, and the kind of like, say you're trying to balance this pathogen control with your selfish gene drives, right? If ideally you'd stay healthy to the very last moment possible when the pathogen kicks in, and then rapidly self-destruct to stop the spread, right? Because with age, you're getting older, you're getting less likely to pass on your gene. Why not keep all that and just shut it down right when you need to?
No, I think it's some animals do that. So like this Semilparo species die rapidly and sharply, but I think that if you have a mechanism of self-destruct, so this red button can be pressed on occasion, and it is dangerous to have it. So you prefer like a more reliable. So this is an interesting question. It's more mechanistic. I'm not sure I have a definite answer for that, but I think the main idea would be that in this model, as Josh mentions correctly, so only chronic sterilizing pathogens play a role. So all the other pathogens will not affect evolution of aging. So, and immune system probably cannot discriminate between the two. So it can see new antigen, but it cannot PCR Google it and predict the outcome.
You could argue that it could evolve that capability. Given evolution.
So the variability of pathogens is very, very high, and they evolve very rapidly. So I don't think it is feasible. So and if we cannot say it is HIV or syphilis that worth self-killing, or it is a flu that will go away in 2 weeks, so then we cannot run this adaptive suicide. This immediate self-killing, which is probably possible only in mole rats. and the bacteria. And actually, in bacteria, most of the pathogens are chronic. So because bacteria are not very good in cleaning out the, so probably, we have papers in preparation for like 2 years. So that indeed the composition of pathogen mixture the population is exposed to sort of modulates the trajectories of aging.
This is exactly the type of discussion I wanted to generate. So, yeah, thank you all for your questions. Let's thank Peter again.